We are starting with section 6.3, and we're going to be talking about the nucleus and the DNA. So the DNA, of course, is housed in the nucleus of a eukaryotic cell, and the role of DNA is to code for it. Pieces of DNA contain the code for individual proteins. So ribosomes make proteins. So what ribosomes do is use the information from the DNA to make proteins. So when we say DNA codes for our traits, what DNA really does is codes for specific proteins, which do determine our traits. We said nucleus contains most of the cell's genes, and it's usually the most conspicuous organelle. That just means it's easiest to see. Okay, so the nucleus is typically the most conspicuous or easiest to see of all the organelles. The nucleus does have its own membrane that surrounds it, and it's called the nuclear envelope, which is a double membrane. It has a lipid bilayer just like the plasma membrane. And I'm just looking to see if there's anything I need to, here we go. Here's some words in bold that we'll talk about. So chromosomes are chromosomes basically consist of DNA and protein, and they're packed very, very tightly, and each chromosome has a certain number of genes, and then genes are like pieces of DNA that code for a protein. So if the cell is not actively dividing, the DNA is in the form we call chromatin. So it is DNA attached to proteins, but they're not really packed tightly together to form strands like the strands that we see as chromosomes. So chromatin is a lot more spread out, and you can't really see the individual pieces of DNA under the microscope whenever it's in a form that we call chromatin. So when the cell is dividing, the DNA, the chromatin, condenses into chromosomes. And in humans, we have 46 chromosomes. 23 come from our mom and 23 from our dad. And those chromosomes contain pieces of DNA called genes. So each chromosome contains a certain number of genes that code for specific proteins. Inside the nucleus, besides the chromosomes or chromatin, the nucleus also contains an organelle or a structure called a nucleolus. And the nucleolus produces ribosomal RNA. In other words, it produces ribosomes because ribosomes are composed of ribosomal RNA. So what the nucleolus does is synthesize ribosomes. Okay? Make sure you know that. Ribosomes make proteins either in the cytosol, they're called free ribosomes, or they're attached to the ER. We call that rough ER. Here you can see what a ribosome looks like. It has a large ribosomal RNA subunit and a small ribosomal RNA subunit. And so a ribosome consists of RNA called ribosomal RNA. All right, now we're going to talk about the endomembrane system, and that is you may have heard of a cell like referred to as a factory. Every cell is going to make some kind of products, and products that it uses within the cell itself or products that it sends out to be sent somewhere else in the body. But the endomembrane system begins with the nuclear envelope. Attached to the nuclear envelope is the rough ER, which synthesizes proteins, and then the smooth ER, which synthesizes lipids. And outside of the ER is the Golgi apparatus, and what that does is package up the products of the endoplasmic reticulum. and it packages those products into vesicles. And those vesicles will either take the product to the plasma membrane and release it outside the cell, or it may be something that stays inside the cell. Okay? Lysosomes are special vesicles that contain digestive enzymes. And then vacuoles are larger than vesicles, and they typically store either a fluid, a solution, of liquid, with dissolved solutes, or it can contain solid particles, food particles, for example, or bacteria. In the case of a white blood cell, it can engulf bacteria inside of a vacuole. And then the last part would be the plasma membrane. And vesicles are produced by the Golgi apparatus, and they are used to transfer the components that are made in the ER. Well, the ER transfers those components to the Golgi through vesicles, and then the Golgi transfers those components, packages the products into vesicles, and transports them wherever they go. So let's talk about the ER first. The endoplasmic reticulum has many, many channels. The rough ER is connected to the nuclear envelope, and it's rough because it contains ribosomes. So the purpose of the rough ER is to make proteins. The smooth ER does not contain ribosomes, and it makes lipids. And you can see here's the nuclear envelope with the rough ER attached, and the smooth ER is kind of outside of the rough ER. The smooth ER synthesizes lipids, and you would have a lot of smooth ER in the cells of our liver, because the smooth ER can detoxify drugs and poisons. And the smooth ER also functions in storing calcium ions, and particularly this is going to occur in muscle cells, because calcium is required for muscle contractions. So smooth ER, these are the three functions. It synthesizes lipids, detoxifies drugs and poisons and other toxins, and stores calcium ions. The rough ER, we already said, contains ribosomes. And the rough ER produces proteins and then distributes them in transport vesicles. So distributes transport vesicles that contain secretory proteins. That just means proteins that are designed for secretion. Maybe, for example, this is a cell in the pancreas and it produces insulin. Insulin is a hormone that is a protein. So it's designed to be secreted outside of the pancreas. So a vesicle contains proteins that are surrounded by membranes, and they're transport vesicles to transport those proteins. So the rough ER is basically, you can describe it as a membrane factory for the cell, a factory for proteins. And then up here it does say glycoproteins. Glycoproteins are proteins that have carbohydrate chains attached, and they are typically found in the plasma membrane, and they function as recognition proteins. The ones I told you about that function in tissue recognition, if you have someone who needs an organ donor, and they have to find a tissue match. All right, the Golgi apparatus, what it does is modify the products of the endoplasmic reticulum and package them into transport vesicles. And you can see the Golgi is a system of these flattened stacks called cisternae. And so once the product, in this case the product is the purple molecule, once it gets to the end of those cisternae, they bulge off and pinch off and form vesicles. So this is a transport vesicle produced by the Golgi apparatus. A lysosome is a type of transport vesicle produced by the Golgi apparatus, but it contains hydrolytic enzymes or digestive enzymes for digestion. The enzymes are made by the rough ER, and then they are transferred to the Golgi through those transport vesicles. And then when the Golgi pinches off, part of the Golgi membrane pinches off and surrounds those digestive enzymes, we call it a lysosome. So how lysosomes work is that once a cell, let's say for example it's a cell like a white blood cell that's capable of phagocytosis, engulfing a solid particle like a food vacuole, or in the case of a white blood cell, it engulfs a bacterium, okay? So once the white blood cell engulfs the bacterium by phagocytosis, a lysosome will fuse with the vacuole that contains that bacterium, or it can fuse with a food vacuole, and all those digestive enzymes will then digest or break down what's inside. So here what you see is, here's the plasma membrane, food coming in through phagocytosis, which is when the plasma membrane pinches off and forms a membrane around the food, and it's called a food vacuole. Then you have the lysosome out here with its digestive enzymes, comes along and fuses with that food vacuole, and the digestive enzymes break down the food. A peroxisome works the same way, but a peroxisome tends to break down fatty acids. Vacuoles are large vesicles, and they are mostly for storage. Food vacuoles are formed by phagocytosis. We just saw that. There are also some vacuoles called contractile vacuoles. In those, the paramecium that we saw in the first slide, it lives in fresh water, and so it's constantly taking on water, and it has these contractile vacuoles that pump that excess water out. And then plant cells have those large central vacuoles for storing water. And you can see that in this image here, plant cell and its large central vacuole. So vacuoles are just typically larger. There's an animation on the central vacuole in a plant cell, and you can find that in section 6.4. And here's a review of the endomembrane system. You have the nuclear envelope, and then you have the rough ER that makes proteins and the smooth ER that makes lipids. And the endoplasmic reticulum, or the ER, will package its products into a transport vesicle. And that vesicle will fuse with the Golgi apparatus. The Golgi will modify and sort the products from the ER. And then they will be packaged into more transport vesicles. And they can be sent outside the cell through exocytosis by fusing with the plasma membrane, or they can stay inside the cell. For example, if those vesicles form into lysosomes, if they have digestive enzymes, then those vesicles are actually called a lysosome, and it will stay inside the cell. There's an animation on the endomembrane system also in section 6.4. Now we're going to move on to 6.5 and talk about the organelles that are responsible for energy, that have to do with energy. Mitochondria produces energy in the form of ATP, and that process is called cellular respiration. It takes glucose from the food that we eat in the presence of oxygen, and it during that reaction called cellular respiration, ATP is produced, and ATP is the molecule that our cells use for energy. Chloroplast, so mitochondria are found in all eukaryotic cells, even plants, but chloroplasts are only found in plant cells and algae, and they carry out photosynthesis. So they will use sunlight energy to convert carbon dioxide into food for the plant. And then there are peroxisomes, which we've already talked about, that function in breaking down fatty acids. There's an animation on the mitochondria in section 6.5, and there's a little bit of information about chloroplast and how they carry out photosynthesis. Chloroplasts have structures inside them called thylakoids, which contain the green pigment chlorophyll, and the stroma, which is the fluid outside the thylakoids. And they carry out the reactions of photosynthesis. There's an animation on chloroplast also in section 6.5. And here you can see a chloroplast. The thylakoids are the green structures, the little green disc. And then the stroma is the fluid outside the thylakoids. And you can see the entire chloroplast has a double membrane, and so does mitochondria. All right, the organelles called peroxisomes are specialized for breaking down fatty acids primarily, and they form hydrogen peroxide as a byproduct, which is toxic. Again, peroxisomes break down fatty acids. In liver, they help detoxify alcohol, in addition to the smooth endoplasmic reticulum, which helps detoxify drugs and poisons. And we will start the next recording on section 6.6, and we'll talk about the cytoskeleton of the cell.